we start?
Yeah. Why don't we start?
So, welcome to all of you are welcome to UL Space Club webinar session 45. First of all, extremely sorry for not switching on my camera. I'm having few issue with my network, so sorry for that. Once again, welcome to all participants and all ISRO veterans and other expert persons all over the country who have joined for our program. All of you are welcome from UL Space Club. So, once again, we are on the 45th session of our webinar series. We started our webinar series in May 2020 during the COVID pandemic issues. So we could successfully complete 44 sessions, and today on the 45th session, we have an interesting topic and an eminent personality to discuss it. Format we, a group of students in UL Space Club, is especially really excited into this particular topic, and we are eagerly waiting to hear it from you. Once again, all participants are welcome to the program. Before going to program or before going to introduction by Shajil sir, I just want to mention few more things that we could successfully complete our National Science Day celebrations in last week. UL Space Club has conducted National Science Day celebrations. We could conduct a webinar and essay competition along with a quiz competition. So the results of quiz competition is already ready, and the evaluation process of essay competition is ongoing. So I would like to declare the first prize winner of declare the name of first prize winner of National Science Day quiz competition conducted by UL Space Club. It was conducted through Google Meet, and the quiz master was one of UL Space Club student coordinator Sri Ram. And the first prize was won by Eldo Sam Varghese, who is a 12th standard student of GHSS Madapalli from Vadagara, Kozhikode. So congratulations to Eldo, and other details of other winners and who the details of students who won the certificates and the details of finalists will be declared soon through the UL Space Club platforms and website. The details of essay competition also will be declared soon. So our last program was conducted successfully, and the details of winners of last quiz program also will be announced in this program. So today we are having an interesting session, like I said. So without any delay, we should move on to the topic. So I would like to request Shel sir to give an introduction about our speaker and this topic. Shel sir, over to you.
Okay. A very good afternoon to all. And we all are sitting for the past more than 2 years under the umbrella of UL Space Club, and it was founded by our Honorable E.K. Kutty sir in 2016, and we are conducting a lot of programs. And the COVID made all the people bring together under the network of these technologies, and we are sitting all the second Saturday. And it is a 45th webinar conducted by UL Space Club to nurture our students. Now there is no geographical limitation to this nurturing program. We have participants from all over part of India. And in addition to that, we are live streaming these programs to the UL Space Club YouTube channel, and it will be recorded forever as a repository of knowledge and the experience. And in the 2 weeks back, we have conducted on 28th of February, we have celebrated the National Science Day. On that day, we had Dr. Y.V.N. Krishnamurthy, and we honored a scientist from NIT Calicut, Professor Dr. Ravi Verma, and most of the scientists were there and the students were there, and just to commemorate those events. And we are proudly sitting here on this event, and today we are discussing about the origin of life and the search for extraterrestrial microorganisms in the space. And we are all concerned about the origin of life and is there panspermia or the chemical evolution still persists there. And in addition to that, we are taking an interdisciplinary approach. And usually the space and all these technologies are the background with physical science or something like that. Nowadays, the world shows that there is no demarcation between all these things. And we have a biological expert, and he's a doctor. And today we are presenting Dr. CBS Dutt. He's a veteran scientist from ISRO. He was a former deputy director of science in Earth, climate, and science area in the National Remote Sensing Center, NRSC, ISRO, Bangalore. And he was contributed to this nation through this organization. He has connected this high branch of science with the life, because the utmost aim of all these technologies to ease the life of human beings and to ensure the existence of life on this earth. That's why we're so curious about the origin of earth and how does it happen and what would happen, how long we could survive there, and all these thoughts were discussed. And that was also an application of space science under the ISRO. Then I proudly welcome and introduce today's honorable guest, Dr. CBS Dutt, and he was the former deputy director of ISRO and in the Earth and Climate Science area. And now he is presently working as an environmental assessor of Quality Council of India, as well as the expert committee member of various research committees of MOESS Government of India for INCOIS and NCOIR and NCESS, and served as a visiting professor at Hyderabad Central University and Gadaj Agriculture University, and extensively work experience on institutional coordination and the program planning and review of projects of a large-scale Indian context. He is an expert in climate change, natural resources, high-altitude balloon experiments of origin of life, and EIA, EMP, and environmental assessment, etc. Successfully coordinated an international ISRO ZNES megatropic satellite program. He has practical experience in executing national projects at ISRO. Currently executed few projects on carbon sequestration in plantations and also in the forestry sector. And is postgraduate from Maharaja Sayajiro University, Baroda, Vadodara in Gujarat, and his PhD is in studies of chemical systematics of Fabaceae and Lamiaceae, the two important major families of the plant kingdom. And he has guided almost 4 PhD theses, and all these are related to this kind of natural importance. Ecological zonation of forest of Western Ghats and mangrove habitat evaluation in Tamil Nadu coast, and estimation of net primary productivity in Indian tropical evergreen forest, and phytodiversity assessment of tropical rainforest in North Andaman using remote sensing. And these are the project and PhD thesis guided by these eminent scientists. And he has demonstrated skills in research and scientific programs of development in the area of earth and climatic science, satellite remote sensing, and effective implementation of large-scale projects through participation of several institutions in the country. The core expertise covering satellite remote sensing, experimental land surface, atmosphere, and coast ocean productivity process of studies. Adequate skill exists in the development of climate-related research through multidisciplinary, multi-institutional participation very successfully. And so that's the importance of today's guest, because he's an interdisciplinary person who connects science to the all branches of science, and he has work experience as a deputy director, ESSA, program director, NISC, and Department of Space, and he was deputy program director in ISRO and program coordinator, Megatropics, and project director on Astrobiology balloon experiments. And he was the group head and forestry and ecology, NRSC, and at the same time he was with an experience of scientists in this great organization of India. And he contributed a lot to this great organization, and he connected the world of science to this technology of space and knowledge of space. And we, the Dual Space Club, proudly welcome you, sir, to this forum. And I welcome all the veteran scientists of ISRO, the teachers, students who gathered here and who join here, who are watching this program through YouTube channel. And a warm good afternoon to all. And without taking much more time, I invite our honorable guest, Dr. CBS Dutt, to deliver his wonderful. Welcome, sir.
Yeah. My first slide is visible, Sajil Saab?
Yes, sir. It's visible. Please.
Yeah. So at the very outset, I profusely thank having considered me as a befitting official for your UL Space Club to my senior officer, Sri E.K. Kuttisap, for giving me this opportunity. At the same time, I also profusely happy to interact with all boys and girls. In fact, it is easy to talk to Professor U.R. Rao or Dr. Kasturi Rangan on this high altitude balloon experiment. But I tremble a bit when I talk to these students, because the type of questions they ask, probably it will not be able to be answered by me at one instance. Now, before I start my presentation, I would like to share to the students of Kerala that we are together trying to learn how is this experiment has been conducted and what are the intricacies that are embedded in it. You may kindly recall this experiment was conducted in 2005, April, from the Tata Institute of Fundamental Research Balloon Facility located in Hyderabad, and it is supported by the preeminent molecular biology labs called CCMB and NCCS Pune. These two institutions have been tasked for analyzing the high altitude air samples which were given, and eventually they could able to detect some kind of microorganisms. Now, having said that, 2005, you may kindly recall, I am talking after 18 years after the experience. So some of the great science aspects, it is difficult for me to embed in this 1 hour talk. So straight away, before going to that, we all know, my dear boys and girls, and you're all my friends and my guests also, you may kindly look at this diagram. It broadly tries to give you, when you look up into the sky, up to, let us say, 120 kilometers, our troposphere, that is where the Earth's atmosphere is, up to 12 to 14 kilometers. Now, as we go further from the troposphere, we will encounter another layer, atmosphere layer. It is called stratosphere. So it will be extended up to 50 kilometers. From thereafter, it will further go up to mesosphere. It will go up to something like 90 to 100 kilometers. From the mesosphere, it will further go up to several hundred kilometers. It is called thermosphere. Now, these spheres are primarily categorized because of the pressure and temperature variations. Now, for example, if I go from the surface of the Earth to as high as 12 to 13 kilometers, my temperature would be something like 8 degrees. Okay, that is how the temperature will fall. No, temperature will be minus 45. Okay, now if I further, what will happen from troposphere, if the balloon is continuously going, it will slowly getting heated up. There is a thermal heating takes place because of the temperature increase, and the balloon altitudes presently recorded in the history of science experiments, it is recorded until 2014 is up to 42 kilometers. That is where the current experiment is also has achieved. So it is at that temperature, the temperature at that altitude, temperature recorded was minus 10 degrees. Now from there onwards, of course, balloon designing needs very large balloons, runs into few lakh cubic meters. So that is what it is not contemplated all over the world for various obvious reasons. So that is how we must really think about the atmospheric layers and the temperature pressure relationship. This is what it is required, because this slide of atmospheric structure and temperature and altitude and pressure relationships, I try to put it here because in the subsequent slides, we may have to use this for ideal gas equations. Now, this is very important. You must understand it's a kind of a philosophy, or as it is a fact. Now imagine the first slide, the topmost small one. I'm trying to look at a picture at 1 centimeter height from a leaf, 1 centimeter height of a leaf. Now, if the same thing, if I look at it 100 kilometers altitude, we will be able to see much larger area, but the same leaf and its aspects are not very much visible. But that means as I go higher and higher. So I'm taking you to the cosmos, then macrocosmos, and then we will come back to the microcosmos. Now, if I further go to 10,000 kilometers, I will be able to see the entire the northern hemisphere. This is where a small circle is there. Now, if I go further higher and higher, up to 1 million square kilometers, you can see our Earth is like a small speckle. And the entire white line is the orbit of the moon. Okay, that is the orbit of the moon is seen. Now, if I go further to 1 million light years, because I have cut short in between several slides, because it's only a kind of a context. So at 1 million light years, the entire galaxy, that our Milky Way is seen, and in the edge of the Milky Way only our Earth and the solar system is there. You can imagine if the cosmos is how big it is. Now further go to 1 light years, the entire solar system and the Milky Way is again becomes a dot. So it is as we go higher and higher, the entire solar system itself is a speckle, and entire galaxy itself will have about 1 million stars itself will become a speckle. Now if I go to 100 billion years, entire solar system, again I'm coming down here, this is about 100 billion years, is 1 light years. Now entire solar system surrounded by the twinkling stars, and several galaxies are seen. Now if I further come down to 10 to the power of 13, 10 billion kilometers, again we will see the planetary movement of our solar system. That is how the scale of operation as we go higher and higher. Now here in this context, we have taken you to 1 million light years, where entire cosmos is seen, where several galaxies are seen. Okay. Now on the contrary, coming back to the leaf, from 1 centimeter to, if I go to if I go to my 10 to the power minus 2 centimeters, you can see the structure of the leaf. Structure and venation, everything is seen. If I go to 10 to the power of minus 4, means I'm going with a high resolution, we will be start seeing the cells. That is where through microscopes and all that we will be able to see. If I go to 10 to the power of minus 7, this is what it is called nanometers. So we will be start seeing the chromosomes. Okay. Now if I further go up to angstrom level, that is 10 to the power of minus 10, obviously the entire electrons which are encircling in a particular cell, they are all seen. You may see as we go.
Excuse me, sir.
Hello.
Excuse me, sir. I have a. Can I ask a doubt?
Yeah. Maybe can we
Sir,
take?
how do we calculate the 10 to the power minus 7 and
Yes,
10 to the power 5? How do we calculate that?
Yeah.
Excuse me. Excuse me,
can
Archit, you be curious till the end of the talk and you write the question there. There will be a question and answer session. Be here and we will be with you. Okay. Don't worry, sir. I think that's a good idea.
You have asked a nice question. Kindly bear with me, my dear friend. We will take up towards the end. Okay. So we know what we are talking is called electron microscope, nano sims, like that. Okay. Ordinary microscopes like that. That is what we are talking. Now, having talked about the macrocosmos and microcosmos, they appear same. That means they are all at the atomic level. Both coincides at the atomic level, but are they same? Atoms are same, but those two are not same. So you must understand, in order to understand the the cosmos or cosmologists, what they are doing, they have launched recently the James Webb Telescope. You all may be knowing. It has been slated at L2, that is something like 5 lakh kilometers altitude above the Earth. Okay. Now, on the other side, because they wanted to go deeper and deeper, because space and time gets unified, but we do not know at what extent the universe is extended, because it is unexplorable depths of the universe. Similarly, if we wanted to go further down, we have been also conducting the Large Hadron Collider, you know, God's particle, what people are talking. They wanted to split the entire electrons and protons further down by the electron bombardment, and they wanted to identify what is further it is possible. It is something similar to the macrocosmos and microcosmos. This is where the sciences, they eventually probably answer what it is. Now, in fact, this is the one which is evading the science during the last 100 years, including the Einstein and all. Now, let us discuss. We have been talking about the origin of life. Now, what is life? Is it a breath of God that gave from his breath in the form of a dust, all these spores have come out? Because it is in a philosophical parlance. This is answers by the very famous molecular biologist, and it should be harmoniously working with an intricate biochemical engine. Because, you know, every human body or every cell, biochemically it works automatically. It's like a micro robot, and which is unimaginable accuracies are there. And another important thing is it's self-replicating, free living, and there is also a guided principle of organic chemistry. So these are all very, very difficult to define and codify how to define the life. Okay. Now, having discussed about some of the challenges on the conjectures on life, every life phenomenon will have a growth, will have reproduction, it will have its motility, means movement, it will have its adaptability, because, you know, humans cannot live in the minus temperatures, humans cannot live in the thermosphere. So this is called adaptability. In every habitats, certain organisms only will live so the life is adaptable. Now, another is called consciousness. Even the bacteria or a virus or COVID-19 will have its own consciousness. It will have its own self-integrity of its own biological system. These are all certain characteristics that need to be defined to call it as a life. Okay. Now, imagine our Earth. We will come to that. The Earth has been formed at the time of the primordial formation of the Earth. Only carbon dioxide, nitrogen, water, carbon monoxide, hydrogen, and hydrogen sulfide were believed to be existing. And believe there is no oxygen at that time. Now, only the energy that was available is the radiant energy. That radiant energy is coming because when there is no atmosphere, entire solar system or the extraterrestrial radiation which is impinging on the surfaces of various planets, that is only the radiant. Another is wherever some continents or the Earth is formed, geothermal energy. You know, in the initial formation of the Earth, Earth's magma was under several hundred degrees temperature. It's boiling. That is called geothermal energy. Another is during the formation of the Earth, because of the Big Bang, so many electrical discharges were happening. So these are all various forms of energy. How come these forms of energy? Because, you know, these energies are required for the various molecular atoms to split them and to form them into ions, and permutations and combinations may eventually may form a kind of an organic matter. This is a conjecture, okay? But some kind of an experiment have been done that I will be telling you in the next few slides. If you look at the elementary table, you can see of the 118 elements, the green things are the essentials for life, you know, carbon, nitrogen, oxygen, phosphorus, sulfate, and hydrogen. Without this life on Earth, it's very difficult to survive. Another is there are some major ions and there are some major transition metals under various separate organisms. There are about 7 lakh organisms, including everything. They all will have one way or other unique to each one of the organisms. Okay, this is where actually the elemental chemistry plays a vital role. So the humans, I will tell you human how much is the carbon ratio. Okay, this is what it is I'm telling. Now I'm trying to gather some information with respect to hydrogen, helium, carbon, nitrogen, and oxygen. If you look at the sun hydrogen is 93%. Okay, oxygen is 0.06%. Whereas in the Earth's atmosphere, we all know hydrogen is only 2 to 3%, but oxygen is 21%. You all agree that the predominance is the nitrogen. Nitrogen is 78%. Okay. Now coming to the bacteria, you can really look at it. Bacteria, hydrogen is 63%, oxygen is 23%. Look at the divergence. Nitrogen is only 2%. Now coming to humans, hydrogen is 62%, is equivalent to that of the bacteria. So what way life of a bacteria, microorganism, to that of a macroorganism like humans? It is almost equivalent, but there is some divergence. Okay, now coming to the oxygen, it constitutes about 2%, but the nitrogen is only 1.1%. So these are all certain things that need to be understood. How on these molecular makeup has made each life system is unique for itself. Okay, the hydrogen now this is how actually it is being done, and we all know hydrogen, oxygen, carbon, and nitrogen provide well over 99% of all atoms in a bacterium and humans. That is what I was trying to tell now. Now, especially when a DNA, deoxy nucleic acid, it is a double helix that I will be talking a little later. It is constitutes a sugar with a pentose sugar, and in the carbon numbers, when we do, this is called first carbon, this is called first, second, third, fourth, fifth. Okay, this is how the carbon atoms are done, and each of the carbon atom is attached with certain kind of amino acids. Now, if amino acids are the building blocks of life, now does the atmosphere has got the primordial atmosphere has got the amino acids? This is what we have been talking now. Now, the left side cartoon is the DNA double helix. DNA is a double helix. Without this, human chromosomes cannot be formed. Okay. On the other hand, if I split the entire thing into a much larger picture, now each of the double helix is interjoined by the essentially 5 amino acids. These what are called adenosine, thymine, cytosine, guanine. Okay, these are all the acids, amino acids. They join with the peptide group, and each peptide group will be linking with the 3rd position and the 5th position. In fact, these two are interchangeable. They are not interchangeable. Okay, this is where our PCR technique uses and exploits the third and 5th band position. Okay. Now, having understood the molecular structure of a DNA and the fundamental blocks of amino acids, now we should know astrobiology. Means in the cosmic area. What is that astrobiology? It is a study of living universe, because the entire universe is not living. In 99.9999% is dead. Okay, maybe 1.1%, 0.0001% may be living. That is what is the cosmos, entire universe consists of. And assess life on Earth in a cosmic context. As I was trying to show you, 10 to the power of 22 level, galaxies are becoming speckles. Okay. Now, NASA has instituted an astrobiology institute established in 1998. Please remember, it is only about a couple of decades back only, has a mission to study the origin. So far, the origin is not known. Evolution, distribution, and future life on Earth and in the universe. If the colonization of Mars or the moon, if it is to be achieved, we have to first understand these fundamental aspects. How does a life consist of, and what are these adaptability? Okay, now the roadmap of Astrobiology Institute, because no space departments in the world, there are about 5 to 6 space agencies. How does life begin and evolve? Because it is a very highly unconvincing to various cosmologists and astronomers. Does life exist elsewhere in the world? Because we don't know Earth-like systems, if they can support the life, is the one which people have been really looking. Now, what is life's future on Earth and beyond? In fact, we don't know. Does the humans are going to live as we are? Are we going to evolve further, and can we adapt? The increased temperatures, increased radiation, and so on and so forth. In such of those conditions, how do we evolve, and how our forms of features will change? In fact, these are all certain things being worked out internationally. Now, just to come back to the Earth. You know, we all know. We don't know. There is a Big Bang. It's believed to have been occurred around 4.8 billion years ago. Big Bang means there are two extremely low pressure masses have collided each other, and the splinters have spread all across. And the amount of radiation that has evolved, it took several billions of years to Earth to cool and the planets to form. Now, if we really see, it is believed the oldest indirect evidence of life is about 4 billion years back. That means it is in that atomic world, in that extreme temperatures and radiation, it is believed. You may ask me, how is it possible? I will be telling you. Okay, the oldest rocks also have been found to be around 4 billion years ago. There are several places across the world, including Australia and America, oldest things were available. The oldest mineral called zircon. Zircon is around something like 4.6. No, it is about 4.6 billion years back. Now, oldest fossils have been recorded around 4.4 billion years back. Now, coming to the continents formation, 3 billion years back, continents have formed. Now, if we further go, how does these life has evolved, and how the microorganisms, complex organisms have evolved? It is a big challenge to the science. Now, one important, my dear students should know, the first and foremost primordial life from the rocks is called stromatolites. These are called cyanobacteria. The primitive microbial organism. It is an algae. It is a calcium secreting bacteria, because it has to live on some rocks, because at that time only rocks were available, and stromatolites have been extensively available in various forms in the beaches of Australia. Now, before we talk, I will not get into too many details. The cells, what we call as prokaryotic cells. Prokaryotic means there are single cells with no clarity on the nucleus. There is a nuclear membrane. There is no nuclear membrane, but it has got a nucleoid. A nucleoid type of thing is there, and it will have an undefined cell wall. It is an undefined, and it will have a flagellum. Okay, not necessarily all the microorganisms will have flagellum, because all the balloon experiment organisms doesn't have flagellum. Okay, they are bacteria, because they are slightly evolved than the prokaryotic cells. Eukaryotic cells are the highly evolved plant cells, human cells, animal cells, and such of those things. And these cells will have multiple organelles within the cell structure, and it will have a well-defined nucleus, and very well defined enclosed nucleus, mitochondria, ribosome, DNA, RNA, all such of those things will be there. That is how the basic units of life have slowly evolved from a single cell to multicellular organisms. Okay, now if we really look at the dimensions, just to give a cartoon, the smallest molecule, that is what we call 1 angstrom, or one molecule is called 1 nanometer. Now we all have come to know the N95 mask we use, because it is a 0.9 is almost near to 1 nanometer molecule, because the smallest of the smallest virus should not enter into our nasal vessels. That is how we use N95. So virus dimension is about 10 to 100 nanometers, because if I use 0.95, that means about 10 nanometers, it automatically blocks the virus. Now, bacterium sizes are 1 micrometer, whereas animal cells are 10 micrometer and plant cells are 100 micrometers. In fact, it is, of course, there are plus or minus sizes are there. Now, one of our friend has asked, all these things are possible through scanning electron microscope and also various staining microscopes. If you really look at it, electron microscope can identify these things from 1 angstrom to as high as 100 microns. Whereas simple light microscope can give you from 1 micrometer band to 1 mm size, ordinary your lab microscopes. So that is how, in fact, the dimensionality of the various microorganisms are seen. Of course, various bacteria and viruses will have different shapes that we will not talk. So if you really look at it, very recently I tried to capture, if you look at it, all taxa, there are 728,103 taxa are well recorded with their DNA structure, and all the base pairs have been listed. Now, if you really look at it, archaea is the most rudimentary primordial type of microorganisms. They constitute around 1,000 available in the world record. This kind of an information is available to the public. Okay, whereas a bacteria, known bacteria, which were labeled very perfectly, is around 34,000. Whereas eukaryotes means like humans, plants, everything like that, there are about 6,83,000 of the 7,28,000. That means all the eukaryotes, all the evolved plant systems, everything, they constitute something like 85% of the whole kingdom. Whereas the viruses constitutes about 8,622, whereas the bacteria is only 34,000. So viruses are very, very difficult to map them, but these things have been very, very well evolved. So from the year to year, these numbers keeps updating. It is as on updated 2022. Now, okay, having listened to the dimensions and the possibilities of the earliest time frame and all that, there are 7 theories on the origin of life. I'm not a cosmologist. This is how no cosmologist will know everything of all the 5 theories. Okay, so this is the fundamental thing that what we have to see. One is called panspermia, another is called simple beginning, another is called RNA world, another is called chilly start, means the deepest cold situations where the Vostok cores in the Arctic and Antarctica, which they have done, this kind of things where in the deep ice cores which is available. Another is called deep sea vents. These are called extremophilies. Another is called community clay. If we keep certain kind of a straw or anything for with the available moisture, automatically straw will generate multitudes of organisms. In fact, this kind of an hypothesis and conjectures have not stood test of time. Then electrical spark. In fact, there are people who try to prove all these things, but of all the things, panspermia is believed to have been gaining popularity across the world. And believers, in fact, we were supported by Professor Jayant Narlekar. He, along with Fred Hoyle, who postulated the theory of panspermia. And as part of that program, when he is approached Chairman ISRO, that that aspect experiment has been conducted. Now, what is actually panspermia? It is basically a cosmozoic theory. What is cosmozoic? From the meteorites and asteroids, when they enter the planet Earth, they bring the primordial spores, the seeds for the evolving the life. In fact, this theory is called panspermia. It is otherwise also known as cosmozoic theory. It is based on that theory. We try to launch a high altitude balloon at 42 kilometers and try to collect the samples uncontaminated. That is one of the challenge which they have given us. We could able to successfully do it, and we found which are not available on Earth, but it is believed to be known organisms in the stratosphere. That we will be discussing towards the end. Now, different theories of origin of life. One is called spontaneous origin, because at the time of the Big Bang, what will happen is so much of ions, atomization, and these things, permutations and combinations can evolve spontaneously, certain kind of a life-forming molecules. Eventually, they have the capacity to replicate themselves on their own, and a life that would have evolved. Another theory is a matter like straw or mud. If we keep it for a longer time with the available temperature, that is what is called radiation or heat, it eventually try to bring up some spores which are hidden in it in the form of certain complex life system. Of course, these things doesn't hold much scientific evidence. Now, there are other 4 earliest theories on the origin of life. One is called theory of special creation, abiogenesis, non-living organisms. That means from the rocks, probably it would have been evolved. It is called mineralogenesis or theory of spontaneity. Then another is called autobiogenesis. Means automatically some kind of a life systems would have been evolved for unknown reasons, because there are certain theories. Okay, another is called biogenesis. Biogenesis means from there was existing certain biological material, but that was dormant, and from that the dormant stage of the cosmic times, it must have sprung up the life-forming molecules. Now, I will not discuss details about this. Now, what is the theory of panspermia? This is what fundamentally rooted this experiment to be conducted. Now, imagine the heavy bombardment or the meteoritic falls or the comets, when they enter into the Earth's atmosphere, they burn off and become atomized into smallest of the smallest molecules. And these molecules, when they enter into the atmosphere, compounded by the initial stages of the Earth's atmosphere, where there is a methane production as well as the various hot springs, hot spots, volcanic eruptions, all have compoundingly provided a lot of energies. So these energies, in addition to the panspermia, the primordial soups, have entered into the Earth's atmosphere way back from 4 billion years to as high as something like 1 million years. Until such time, continuous spray of the primordial things are happening on Earth's system, and that is how the complex molecules have evolved on the Earth. That is how the panspermia theory illustrates in a brief sense. Okay, this is how the molecules must have been evolved. A dense cloud means cosmic dense cloud. Okay, then there is also a diffuse radiation, and there are some stars will die. That is called stellar death. Eventually, when these kind of star and planetary formations are happening, excessive exogenous delivery will be there. When there are some deaths and there are some star formations are there, all these things will deliver so much of comets and interplanetary dust particles. These are called IDPs. These things will continuously flow onto the planet, and these things are believed to be the primordial spores in forming the Earth's life. Okay, this is one Michael Callan of Goddard Space Center. This is one of the oldest rock of 4 billion years old, where it is believed these kind of things have been formed in the primordial stage. Okay, one of the important thing all the my friends of students should know, one of the important thing is called Stanley Miller or Uri Miller experiment. So what he did is he had mixed the primordial molecules like methane, ammonia, water, and hydrogen under the controlled pressure of the primordial earth, very, very high temperatures. He could able to synthesize certain kind of molecules like, amino acids. So the formation of organic molecules, including several amino acids, but not the life. Okay, but this is one of the strongest experimental evidence called endogenous synthesis from primordial soup. So this kind of things would have happened at the time of the Big Bang, when the cosmic dust, when it is continuously forming, star death is happening, stars explosions are taking place, comets are falling, all these things under the excessive radiation scenarios, probably this kind of likelihood scenarios would have happened, and eventually life would have been formed. However, the Stanley Miller experiment did not find the formation of life, but the building blocks of amino acids have been detected, including the few organized organic molecules. That is one very important takeoff from his experiment, but this was done way back in 1953. Okay, now, interstellar clouds will have a very ionized species like hydrogen, cyanide, hydroxyl, methyl group, and silicon. Okay, these few complex polycyclic aromatic hydrocarbons called PAH, these things are in the gas phase. Okay, now these molecules in the molecular reactions in the prebiotic atmosphere may have synthesized the formaldehyde. This is a very, very important thing. If somebody wants to read the chemical evolution by Melvin Kelvin, in fact, there is an excellent book called Melvin Kelvin. It is in that book, the formaldehyde probably is the most precursor element, along with the hydrogen cyanide, are the building blocks of our sugars, and the nitrogen bases required for RNA. That is riboxynucleic acid. Okay, this is called single strand RNA. Okay, this is how probably these two things would have been formed from the formaldehyde and also the hydrogen cyanide. Okay, then what is that evidence? We have the Murchison and Murray. Murray is in US and Murchison is in Australia, Western Australia. Have many amino acids, purines, and pyrimidines. This is basically a rock. I will try to show you the rock, and this rock has been analyzed by the petrochemists, and they found all these amino acids in that. But this is what is Murchison amino rock. This is a meteorite. It is a meteorite which they have analyzed for all the fundamental blocks. So this confirms the evidence of rocks will have hidden primordial molecules. Okay, Murchison, a town in Australia, a meteorite which was fallen in 1984. It is a carbonious chondrite, remains of fossilized bacteria similar to pedomicrobium. In fact, they have even found certain kind of a fossilized bacteria in such a high resolution. They could able to find. So this is what they could do. There is also an interplanetary transfer material is well documented as evidenced by a meteorite probably believed to be coming from the Martian surface. It is called ALH 84001. It was found on Earth in 1996. In fact, this also has certain rudimentary traces of the microorganisms like features which have been detected using the high resolution electron microscope, scanning microscope, and various chemical analysis. Okay, now I will tell the current thing. This is an inspiration coming from the Fred Hoyle and Vikramasinghe. Vikramasinghe is from in Sri Lanka. Expanded the theory of panspermia. there can be no origin of life from non-living matter in the finite past. Please remember, what he is telling is there must have been in the cosmic past, there must be a living material. But that living material, what we think like a bacteria or a virus or a prokaryotic cell, it may not be like that. It may be in altogether different form. Okay. Now, confining to the various fundamental functions, as I explained in the first few slides. Now, the cosmic ancestry holds that life on Earth was seeded from, say, from space. That means whatever but who are the transporters? The transporters are cosmic, the meteorites and the asteroids and all. They must be bombarding, and they brought the cosmic dust. Okay, the Vikram Singh and Hoyle hypothesized that the most promising location for the synthesis of prebiotic molecules by Miller and Urey, that is what I was telling you. This is what it has substantiated the Vikram Singh's expanded panspermia theory. In fact, the Professor Jayant Narlikar, our own Indian very famous cosmologist, he is a doctoral student of Fred Hoyle, and he and Vikram Singh worked together at the University of Cardiff. Okay, so this I will skip it. At his initiative, this experiment is conducted, and this is a national level brainstorming session by the preeminent officials of the national microbiologists in the country. The best microbiologists are available. This is Professor Rao, this is Madhavan Nair, the then chairman. This is P.S. Goyal, the former the Secretary, Minister of Health Sciences. At that time, he was the director, Isaac, and sorry. This is Professor Pushpa Bhargava. He is one of the most outstanding molecular biologists. He is one of the most mentor and inspired me for conducting this experiment. He is Lalji Singh, the former director of CCMB. He is Professor Shivaji. Of course, he is the director IMTS at Chandigarh. This is Sauche, and there are several people. And he is Professor Mr. Rajaratnam. He was my predecessor who conducted the first initial experiment. Now it is in this experiment. In fact, this is myself, and I request. There are several preeminent people, including Vijay Rahman, Alex, Kiran Kumar. They were all there on the other side of the thing. Of course, at that time we didn't concentrate on the good photography. So at that time they discussed the experiment which was conducted in 2001. It has been severely criticized because it would have carried from the earth along with the dust so many microorganisms, and it is contaminated. So some of the challenges in the national level brainstorming session has educated me to conduct this experiment very carefully, taking the clue from the previous experiment. So what is that big inspiration? Now it has proposed a challenge to the scientific community again by Professor Jayant Narlikar, Origin of Life, and his unstinted perusal for the various experiments since 2001 with ISRO. In fact, again in 2001, when once it was criticized, even in the international scenario, he had asked, why can we discuss and again reconduct this experiment? Now, this experiment in 2001 is one of the most outstanding experiment, and I try to climb in the National Molecular Biologist team and various people. It is on par with the LIGO and LHC, Large Hadron Collider, where we are talking about the microcosmos and God's particle. LIGO is nothing but to look at the large interferometric ground observatory. Now we are talking about the James Webb Telescope to look at the cosmos into the deeper and deeper and deeper depths. Okay, now at that time we have been already lost. Even now unstinted efforts are called for. There is a scientific inquiry and there is no answer all over the world. But we have with us people like Jayant Narlikar, great cosmologists. We should take it further. So before we go further, first understand the Earth's composition. 98% is nitrogen, oxygen is this, and argon and all that. Now here I am trying to talk about the neon. And why is that neon 0.0018% is highlighted here? Because this is the neon which is used for minus 270 degrees cooling, minus 270. Can you believe none of these trace gases will be able to provide except the neon. Why neon? Because neon, of course, before we talk about the balloon, because I have to take the balloon to a height of 42 kilometers. In order to take the balloon height, I request the students should understand the fundamental equations of ideal gas equations. It is called ABC equations. One is called Avogadro law, that is V is proportional to N, and Charles' law is V is proportional to the temperature, and Boyle's law is also V is inversely proportional to 1 by P. So these are all interdependent. And if I wanted to estimate what is the size of a balloon which I would be requiring, I should be able to use this kind of an equation. V is equal to constant multiplied by NT by P. That is how we have used. R is a gas constant. We all know gas constant is 22.7 value. Okay, it's a 0.08. Now the ideal gas equation, if I wanted to really use how much I have to consume and ensure the expandability of the balloon, I must know the fundamental principles of because these are all the constants. P is the pressure. At 42 kilometers, my pressure will be 2. But of course, we will also give a margin of 1. And the volume, what will be the likely volume? And N is the number of molecules. And temperature at that time will be minus 10 degrees. So using all these equations, and R is known already. R is this. R is this 0.082057 liters of atmosphere per mole per Kelvin. So using this equation, it is possible for us to back calculate what is the likely volume that would be requiring, and what is the atmospheric mass and also the molecular mass. Okay. Now let us look at the molecular mass of neon is 20.2. Now why is this we are talking when I try to compare with the helium, it is 4. But on the contrary, we have to look at the other physical properties. Liquid neon is an important cryogenic refrigerant. It has to be super cooled. My instrument, it warranted a super cooled temperature. It has over 40 times more refrigerating capacity per unit volume than the liquid helium. Okay, and more than 3 times that of liquid hydrogen. Okay. That is the one, the liquid neon has been chosen. Please understand this kind of a preliminary understanding has already been acquired from the previous experiments. And also the Max Planck Institute of Aeronomy, which they have conducted experiments for the chlorofluorocarbons at that altitude, not for the biology. Okay. So neon is a noble gas, because it doesn't interact with any of the people, and neon is almost colorless and odorless, and its density is 2/3 of the density of air. Density of air is 1.2 grams per liter, whereas it is 0.9002 grams per liter. That means if I condense this, I will be able to condense more amount of liquid neon than that of the hydrogen or neon. That is the advantage. And my cryogenic cooling will be 40 times higher than the hydrogen or the helium. Okay, that is how we have used. Okay, now you may ask me, how is that we have estimated the atomic mass of neon using the same PV is equal to nRT, and I substituted all these values and estimated the 20.1727 micromoles of atomic moles of neon. But whereas here, if you really look at the density of air is 1.20, and as I was telling you, the density of neon has been estimated as 0.900. Okay, that is what we can calculate from these same equations, ideal gas equations. That is why I request my students, please do not ignore these equations, fundamental equations, because at that time there will not be any direct applications, but as we grow higher and higher, we may find its application. Okay, now why is that I'm talking about supercooling, and how is that the balloon has been taken to such high altitudes, and how the samples are collected? So these samples are collected, each one will have about 900 milliliters, equivalent to 1 liter volume at the surface of the temperature, and this is called cryotherm. In this cryotherm, each of these stainless steel tube will contain 1 liter of air that can be sucked from the standard temperature, and the sucking is done from inlet is only from here, and the any excess gas I can let it out from only one outlet. Okay, that I will be showing you in the expanded image later. Now, likewise, all these cryo probes, these are called, each one is called cryo probe, and the compound module is called cryo module. And each of these cryo probe is operable through a telecommand through a motor. Vertically the motor can rotate left and right, and each one is under the direct control of telecommand and telemetry. Okay, in fact, over a period of 3 years, I have learned all these things, and this is how the cryo module in the open up picture, this is how it look like. Okay, now when a sample need to be taken, what will be the rate at which we should be able to sample? See, sampling rate will fall by 10,000 folds from 20 kilometers altitude to 40 kilometers altitude. So if the sampling rate is falling, I will not be able to get, because atmosphere is so thin, pressure is about 1 millibar. So if the pressure is 1 millibar, my whatever tube I am taking, it will not be able to suck. If I wanted to suck, my pressure should be negative. So we have ensured, using the turbomolecular pump, all these modules have been pre-evacuated to 10 to the power of minus 6 bar. Means from the normal atmosphere, it is minus 6. Now, in addition to that, what we did is we soaked all these things into a cryotherm. This is called cryotherm here. Initially, before the launch, we put it in the liquid nitrogen. So liquid nitrogen can take the temperatures up to minus 160 degrees. But that is not good enough. We want to minus 270 degrees. So we have imported from Ukraine. The present war is going on. Specially specially ordered cryogenic liquid neon and 50 kg. Each 50 kg costed something like 38 lakhs, and it was delivered to Hyderabad. Of course, we have so many problems. So ultimately, we have decided, as the altitude goes up, we know what is the specific orifice that is required in the cryo tube while sucking, and how much time we have to suck depending on the time that is available, like that. Now, you may ask, how is that we are able to get the time? Now, this is thanks to Dr. Balasubramaniam from SPL. This is how the high altitude profiles of the wind speeds. In fact, this is for yesterday. I requested him kindly provide. I didn't have a good picture of my 15 years back balloon data, though I contacted the TAF, Dr. Srinivasan and Sunil Kumar. They too didn't have the digital data at the time. Maybe retravel has become a bit difficult. So using this vertical profile, it is possible for us at 30-40 kilometers, what will be the likely wind speeds? So here in this picture, of course, this is belong to March, but we have launched in April. So at 12 kilometers altitude, winds are blowing with something like 30 meters per second. Okay, winds are blowing 30 meters per second, whereas the high altitude winds are as low as 6 meters per second. But this is not the case. At the time of our experiment, it was something like 250 kilometers per hour. That is how we have estimated. During the experiment, this is the turbomolecular pump, and how is it getting operated, and how is it connected, and how do we disconnect to the cryo module. In fact, the cryo module is this. Okay, this is the one which is connected to the turbomolecular pump. And of course, there are 3 times Professor Rao and Narlikar, they visited the TFR balloon facility where the entire cryo module, this is an electromechanical instrument. This has been integrated and successfully done, and it was under their supervision, constant guidance, and Professor Manchanda and entire staff of TAFR, they were with us and we could able to do. In fact, the entire astrobiology payload look like this. It will have telemetry boxes, it will have ballast, it will have the inlet pipe. This is called inlet pipe. Okay, now this is called cryotherm, and just before the launch, we will also undertake certain kind of a high altitude wind sounding. This is Professor Manchanda. In fact, they were giving us certain guidelines how to ensure the contamination issues and all. So right from the day one, the entire instrument has been airtight and sealed with a pyro cutter. In fact, this was sealed, and there was a pyro cutter support was given by VSSC. At 12 kilometers altitude, we could able to cut the pipe so that the entire inlet pipe is open to the cryo module, and it can be with a telecommand, everything can be done. Of course, before the entire experiment is to be take off, we were very fortunate. Dr. Vedachalam, former director of LPSC, he was requested by Dr. Madhavan Nair to overview, and he was with us for 3 days. And believe me, he was overwhelmed with the basic science. Though look simple, very complicated, and he patted me so many times, and he took me to a nearby temple, and he blessed me that next 2 days the experiment is going to take place. In fact, he is Mr. Srinivasan. He is the man who provided me the balloon engineering support, and Professor Manchanda is the chairman of the balloon board. He was like a shadow with me, and I ensured that entire electromagnetic period was successful. And there are several other people, and he is Mr. Datta, who supported me from headquarters time and again wherever some technical aspects are required. This is how the balloon launcher. In fact, it's not like PSLV, GSLV. It's like a balloon launcher. It will have the pilot balloons. Okay. Now, this is the payload. This payload is suspended as a mass to the balloon, and it will be taken up higher and higher. Of course, there are certain aspects which have been taken, and the balloon volume is something like 422,000 cubic meters aloft, taking the 675 kg of the payload. So this is how we have taken care. Of course, we have taken the clue from the previous experiments. They were not meant for origin of life. They were meant for the chemistry. So chemistry doesn't warrant the sterilization. We have ensured the pre-evacuated samples at 10 to the power -6, and we could able to successfully do that. Okay, the inlet valve was kept open till sampling altitudes, and need specific biological protocols for sterilization. And payload assembly to be made in a clean room. We have developed a clean room with ultraviolet rays and all that. Okay. Of course, there are several hundred things. New improvisation techniques have been done. The pyrotechnic has been used to open the valve at 12 kilometers altitude for the first time in the history of, not even with the Max Planck Institute, thanks to VSSC pyrotechnic group, where the temperature was -50 degrees at 12 kilometers. We could able to cut open and ensure that there is no terrestrial contamination that is carrying the dust. Of course, the entire sample assemblies were personally handed over to CCMB, and we have also ensured the cryo module, the temperature monitoring on the ground. There is also a monitoring station that is where we have done, and we have improvised the whole things. Okay. I will skip on the 2005 salient findings. Air samples did not yield any bacteria or fungi except in one sampling tube NCCS, possible reason. Okay. Washings of cylinders did yield bacteria and thing. See, if I take -10 to the power 6, the entire air is liquefied. Okay. And of course, these are all the things, preparations going on. This is the command control. The entire command control on operating all the 16 valves at specific time, specific altitude, specific time, and opening the orifice of the Nupro valve is commanded by me, because entire thing, go or no go, has to be by one specific person, because that is me, because I have understood end to end of the various issues, thanks to thoroughly guided by the TAFR team, and there are certain TCTM team, they have done a marvelous job. Now, before that, what we are talking about, we will take these different altitudes. When we operated, it is 21 minutes, and the pressure, the cryo module pressure was 333. Imagine if a sampling tube of 333, it is likely to be burst, isn't it? If a cryo module is burst, entire cryo module and entire experiment will be exploded along with the hydrogen. Hydrogen is a very explosive gas. So we have ensured there is a burst test. Say all the stainless steel tubes have been tested to withstand the 600 bar pressure. Okay, when the air is sucked inside and liquefied instantaneously by the minus 270 degree liquid neon, the entire air gets liquefied. But as we go higher and higher, you may see the liquefied or rarefied atmosphere will have very minimal density. This is where we expect the microbial organisms, and that is what we have successfully did it. Okay, now we have ensured the conformation and closure of 16 microchrons. That is what is required. And all the, after descending the entire payload back on to the Earth, fallen at 250 degrees. This is how the balloon is released. And once the balloon is released, this is called balloon launcher. When it is released, it is, of course, it's a graphically plotted a tree height profile. In fact, it is reported by their records 216 kilometers at Bidar. Okay, it has fallen in Bidar. Now, I must say here, along with the students, one of our senior colleague, Dr. Geeta Vardhan, she was interacting with the various defense officials. Madhavan Nair has requested her to enable to fly from Hyderabad airport and quickly fetch the instrument, and we could able to, this is the instrument which has been landed back after having gone to 42 kilometers. And this is myself and this is my support colleague. We both went into this helicopter immediately after having no sleep for 3 days, because we have to do the launch preparation. And this has been immediately dismantled, because, you know, this should not be tampered and contaminated the tubes. And eventually all these things, you know, these local mob handling has become a very, very tough job, because we don't know the local language and all these things. Anyway, these are all certain issues of the challenging experimental aspects. So eventually we have collected the weight of the pre-flight and the difference in weight, and what is the grams of equivalent water. That is what is called R value. Okay, this is what we could do, and these samples were equitably distributed to both CCMB and NCCS Pune, and these people have eventually found out certain bacteria. This is what is called, each one is a name, each one is a culture number, and each one has got what is called bacteria, fungus, or whatever it is. Of course, all these fungi, even today, they were not immediately identifiable in the Indian context. Okay, now this is what we have done, and we would like to record here the one species gnome in this experiment. Apart from 12 microbial organisms recorded, one was recorded for the first time. He is called species gnome. He is called Cyanobacteria hoyle. It was given, Fred Hoyle is postulated the theory of expanded theory of panspermia along with Norlecker. Of course, it was published in a Nature article, and this was confirmed by DNA hybridization, where the homology is below 95%. This is UV resistant, it is gamma resistant. That is how all these spores must have been must have come from the extraterrestrial source. So these are all certain things which are taken. I will not detail further. And this is the balloon float altitude planning has been done with a theoretical understanding about all these things. Now, having said all these things, these are fundamental physics is required on this. Having said all these things, you must know high altitude balloons has been a great inspiration to our ISRO's launch. In fact, when I was discussing with my boss, Dr. Madhavan Nair, the then chairman, I told him, sir, we have been using for spaceport launches using Sphere, which will go up to 1 to 1.5 kilometers. Why not high altitude wind sounding from TAFR be used such that the wind attacks or wind loading factors up to 30 kilometers can be used for launching our PSLV and GSLV with an improved aerodynamics control. I'm very happy. This is one spin of that one, and TAFR has supported the SHAR Center, and now continuously high altitude wind sounding is done. Concomitantly, the vertical wind profiles which are modeled. That is how by SPL and all that, which is being used. That is how they work together. Another spin of this is you may see this is Mr. Srinivasan, who was with us when I was showing you along with Vedachalam. In fact, the moment he was retired in 2012, he was drafted for the services of Alan Eustace. Alan Eustace. His jump record is the highest in the world because of using the TAFR balloon, and he has jumped 41,422 meters, whereas ours is 42,000 meters, almost same. In fact, he has gone with a standalone spacesuit. You know, he was just looking down, and the entire thing is spacesuit, and it is cryogenically and radiation resistible. It is actually a spacesuit. This is how he was getting aloft while getting. This is a picture I have taken. This is how he was in the spacesuit. This is how he has taken the picture of the panoramic view at 40 kilometers. And he was giving a TED talk on how he has done. And this is the spacesuit. Mr. J. Srinivasan, Mr. Srinivasan from TAFR, along with the Alan Eustace. Of course, Julian Knott is now no more. He is the one who has carried out 90 successful balloon jumps and world records. Of course, he has superseded him, and the entire thing. He is the senior vice president of Google. His passion, that kind of things are required. And this is the spacesuit through which he could able to flow. And this is just at 40 kilometers. He is about to be ejaculated by the telecommand. The entire balloon will get burst, and he could able to descend to the Earth's surface in about 8 to 9 minutes with a velocity capturing up to 1300 meters per second. So that is the velocity at which he has dropped. So thank you very much. And this is all about the great inspiration and experiments which we have done. And this is where my immediate boss at the time is Vijay Rahman, Scientific Secretary Professor Rao. He is the main inspiration to me. He stood like a rock with me. He is the Professor Manchanda, he is Jayant Narlekar. And of course, this is the Srinivasan, Vedachalam. Of course, name everything is all because of their unstinted effort. Thank you very much, and it is open for discussions. Yes, sir.
Thank you very much, CBS Dutt. And it was a really interesting story. As I am also a person from biology background, we are talking to the students about evolution and these things. And it's a clear eye-opening session, and there was a lot of reference also from the biology people. So we really thank you, and the students are so curious about these things. And these things hasn't happened in the US Space Club yet, because we discussed about the other aspects of trajectory or some other propulsion or the satellite, something like that. Then we have been aware of the fact that the ISRO is doing so much of experiments in astrobiology, because we have a girl from the Calicut. She is Arya Raj. He is nurtured by our Ika Kuti sir, and he is a cerebral palsy person. and he want to study all these things. And now he joined in ISRO Trivandrum, and he liked to learn what is, he like to conduct research in astrobiology. So, so the thing is there. So thank you very much, sir. Then I invite DK Kudi sir before we going to for the question and sessions and for the veterans also. Welcome, sir.
Thanks, Shajil Mash. It's eye-opening is the word you used. It is very, very true. Very, very. We are exposed for a first time. Our students are exposed to the first time on such an experiment, and that too, CBS Dutt has taken us in on a path, very interesting path, as if we are ourselves a part of the experiment. The way he briefed, the way he presented, the way he trying to convince us on the experiment is something like we are on a real time. We are also a part of the experiment. That is a kind of ambience created by my dear friend CBS Dutt today, and it is something very, very different from what we have been traveling so far in the last 45 episodes plus 15 special webinars we had for various occasions, including Science Day and all that. So this is a very unique experience for our students, and I don't want to come between the students and Dr. Dutt. They have a lot of things to talk and ask, etc. Even my own veteran colleagues from ISRO who are listening here, some of them may be quite interested to butt in and comment on this. So it's a very good thing what Shajil Mash said about Arya Raj, one of our students who did her +2 and then got entry into ISR. She's immediately when I saw she is not present today, I called her, but her number was switched off. Then I called her father, spoke to him. He said she is in an online class from ISR. That otherwise she would have been there. Even on the second Saturday, she has some assignments and things like that. So she couldn't. Otherwise, she would have been very exciting, and we would have been very happy that she got a first-hand experience from a person like Dr. CBS. Thank you so much, CBS. It's been a very, very fantastic experience and exposure for our students, and it's a very nice experience for me, although I have had lots and lots of presentation like this, but this is something very unique I felt in today. So very, very happy, and thank you for making this such an interesting day and interesting talk. So we'll open the forum for students to interact and worthy comments from my veteran senior colleagues and friends from ISRO here. Thank you.
Sir, first we give the chance to our students, and they may be eagerly waiting to ask some questions of that part. Varun, and who is handling today's question and answer sessions? Sajid?
Yes,
okay.
sir, I'm handling.
Sajid, introduce yourself and continue.
So I'm Vijay Rahman. I'm a NIT Calicut Mechanical Engineering first year student. So first of all, I'm thanking you, Dutt sir, for spending your valuable time for us to give this lecture. It's actually a beautiful lecture, and it's an inspiring session for us, because most of our students are inspiring to get into ISRO or that kind of big institutions to work. So it will be really inspiring for all of us. So thanking you, sir, first of all. And it's time for the question and answer session. We already received some questions in chat session, and there are lots of students have very, very curious to ask you directly questions. So let me first invite Niranjan K. Manoj to ask his doubt.
Hello.
Yes, sir. Yes, Mr. Niranjan.
Niranjan, open your video and ask.
Sir, here I have an issue, sir. I can't open my video. It says that webcam application is blocked.
Okay.
Don't worry. Ask the question.
So mainly the question is, so which kind of early creatures first appeared at the time of the Cenozoic era than the other creatures of that time? We all know that there were three types of era. One is Mesozoic, another is Proterozoic, and this is Cenozoic. So which kind of creatures first appeared in the Cenozoic era?
Yeah, you are talking about the geological eras, Paleozoic and all that. So they are all geological times. The primordial life in the tree of life, it is called archaea. Means the most fundamental rudimentary organisms are called archaea. So that is what it is internationally, uniformly they agree. The archaea forms are the first primary organisms must have been formed. They are all unicellular, or sometimes very difficult to classify them. Are they unicellular at all? Like that. They are called archaea.
Okay, sir.
Excuse me, sir. I have a doubt because I have another class, so I am asking first. So, sir, I have a question. How to count the 10 to the power 23, 10 to the power 24? How we calculate into numbers like billion, trillion? How we can convert to the words, the 10 to the power 23 words into words?
It's a wonderful question you asked, but it is nothing but distance, isn't it? What is 10 to the power of 1 meters? 10 to, in meters, if I want to say 10 to the power of 1 is 10 meters.
Okay, so we want to tens, hundreds, thousands, 10,000. Like
it is
that,
exactly
right?
multiples of tens. That's all. That is how the numbers are given. Okay.
Okay, sir. I understand that. Thank you.
Thank you.
And it was an interesting session. I like that. This session.
God bless you.
Thank you, sir.
Yeah, Mr. You can moderate, Mr. Sajus.
Okay, sir. And those who have doubt can raise your hand so that I can call your name.
Hello, Chandni, you have something to ask?
Yes, Chandni, Chechi, you can ask.
Your mic is off.
Your
mic
mic
is off,
is
Chandni.
Yes, sir. Thanks, Dutt, sir, and all other my student friends and my sirs. So, sir, it was quite interesting topic. Actually, as we student members of US Space Club started searching for what is actually this topic is all about. So, as a preliminary research, we could find something interesting. Give more detailed explanation of all those things that we have gone through preliminary. So, sir, I got interested and we have seen such one thing, that is one U profile. We came across a word called as U profile. Okay. So, sir, it was giving an idea like it was a graph that is a plot basically plotted between the altitude as well as the density of the microbes. So, what is it actually implies? What is the importance of that?
Yeah, see, this is when we are talking about the exogenous theory, there is a constant spray of dust will be there, isn't it?
Okay, sir.
So at different altitudes, at vertical levels, if the samples are collected at a specific volume and specific time intervals, the number density of bacteria that is available in the aerobiology, okay, it comes under a simple aerobiology at different altitudes. So the number density, when it is plotted against the altitude, it is called U profile. Okay.
Okay. Sir, one more question. Actually, we got very interesting information about how the samples, the Cosmo dust is actually collected and brought to our labs. So we came across a pouch that is basically a low density material made or capsule like of thing in which the Cosmo dust is that collected was brought to our labs. So what is the importance of the cavity? What is the importance of that design of the cavity in which the cosmodust captured was carried? And what's the significance of choosing the loaded couch for that?
Yeah, see, that is called orifice. Okay, so the small pinhole. Okay, now pinhole. If I wanted to suck the external atmosphere, if my pinhole is larger, in a short time I will be able to suck the more amount of air. Okay. Now, if the pinhole is smaller, I will not be able to suck the entire sample in a specific designated time, because balloon is keep on going higher and higher with a velocity of 8 to 9 meters per second, because that is what is called float velocity. Okay, these are all some scientific ballooning. So that is what we decide. If it is going much beyond, the balloon will burst. So we ensured by keeping an unnecessary suspended mass, it is called ballast. Okay. Now, if I want to collect between 30 kilometers to 40 kilometers altitude, 2-3 samples, I must decide what is the time at which each of these sampling tubes have to suck the sample. Now, if I put more time rotation, that means more orifice I am giving, more larger space, 0.1 mm to as high as 2 mm diameter. Okay, that control I have depending on the time. That is what it is in the command control room, which we will be able to decide accordingly the volume of air is collected, because by the time it will touch already the undesired altitude of 40 and above. Okay.
Okay, sir. Okay. This is a very quite interesting topic, and thanks for giving us such a nice introduction to such a vast topic. It's quite.
Thank you, sir. So, I myself has a question to you. Can I ask?
Why not?
So, you said your experiment was conducted in 2005. So, what will be the involvement of the latest technologies? Because now computers are so super fast than 2005. So, what will be those involvement if we conduct the same balloon experiment in 2021?
Sir, my dear friend, don't get away by these supercomputers, cloud computing and all. Okay. Now, this has nothing to do. It is imagination and inspiration that has worked in collecting the sample. Now, you can see ISRO after 2005, because we have the greatest cosmologist available, Jayant Narlikar, who worked with Fred Hoyle. Subsequently, there were several efforts. The mass of the payload, we wanted to reduce it to 15 to 25 kg, and several discussions were given. And initial, I know as a non-engineer, I can think because having wetted my hands. So it was not taken up. But there are certain initial plans using the PRL nanosims. A single bacteria, if I am successfully collected, I can identify and do the radioisotopic analysis and ensure that whether it is a terrestrial origin or extraterrestrial origin, because C13 by C12 ratios of the land is different than that of the extraterrestrial ratios. So this kind of things have been planned, but somehow it couldn't take place. But one of the greatest inspiration and experiment by Alan Eustace, he has gone personally himself up to such an altitude. And in fact, this was the experiment designed to ensure our Kalpana Chawla at 80 kilometers altitude, when the carbon-carbon fibers, they ruptured, when the space shuttle is entering to the Earth's surface, she died. There are about six people died, burned themselves. Now, one of the most challenging thing is they can eject themselves at that altitude using such similar space suits. So this is what the Alan Eustace has been working out. So that is how one can save the life. That's why I requested Kuti Saab, if I'm blessed to have Lalitambika's presence, it would have been nice. but ultimately we couldn't get here. Okay, thank you very much. I hope you understood, sir. Sayu?
Yes, sir. So those who have doubt can directly ask. Don't be shy to ask any doubt.
Hello, Professor Dutt. Good afternoon, sir.
Good afternoon, sir.
Myself, R.K. Sarangi from SAC, Ahmedabad. I have a query regarding, it's a very excellent presentation on this high altitude balloon experiment for studying this astrobiology experiment on this.
Hey, can you switch on your camera here? I wanted to see Sarangi. You are our
yes,
brother.
sir. Yes, sir. Yes, sir. Yes, I'm.
Okay. You are showing your steps. Doesn't matter.
Yeah, it is coming here reverse. So
Doesn't matter. You speak. Let's save the time.
Yeah. Okay, sir. Sir, actually, this return sample we have from moon lunar return samples. So, is it any experiments have been done for this microbial study or astrobiology of this return lunar samples by our ISRO community or any other space related institute, astrobiology institute in India?
Are Babre, you asked a very complex question. In fact,
nice, sir.
the planetary mission team,
yes, sir.
They are fully aware. Mr. Goswami is there, and in fact, their team can as well look after. Or they could have approached Director EOS or myself. We would have directed them how that sample can be utilized, because it is already contaminated, because the moment it comes lunar sample.
Yes, sir.
But anyway, certain kind of experiments can be done by rudimentary analysis, even if it is petrified.
Yeah. If no more students are there any more students asking question?
No, sir.
Yes, sir. I am visible, Professor Dutt now.
Yeah. Sarangi sir, namaskar.
Namaskar.
I remember your face.
Yes, sir. I have met you many times in Darjeeling and at NRSC Hyderabad and some.
I remember. Thank you.
Yeah, excellent study, sir. Hope to expect more astrobiology and this samples related and even balloon experiment related study for information.
I retired, that was done in 18 years back.
Yes, sir. Yeah, great. So I think a lot of continuity.
I think we'll give the time to
Thank
students.
You, sir. Yes, sir. Thank you, sir. Thank you. All the best.
Okay. We have with us the most important person who has been guiding and directing and doing a lot of things, Dr. Vijay Rahman. He was also part of it, and he was shown by Dr. Dutt in the experimental lab and all that. So, Jay, why don't you make some comments about what is talked by CBS?
I don't want to add anything more because it was an excellent, fantastic talk by CBS. He gave the fundamentals of astrobiology to the experiment level. Only thing he has to probably forward to tell us the payload weight. Weight of the payload was 850 kg payload, but correctly at that particular point of time.
Actually, sir, to be precise, it is about 625 kg.
625 kg balloon flight. Can you imagine?
No. Sir, total along with the balloon, mass, telemetry, telecommand, electronics, batteries, everything, it is almost something like almost near to a ton, 1 ton.
I remember 850 kg or whatever. So
No. That is payload, sir. That is payload.
Okay. So
payload is actually 625.
One more thing is the 3 bacteria species found out extraterrestrial. One he named as Fred Hoyle, other 2, one was on ISRO, another was Autobiogenesis. Maybe able to mention to the students so that they will be better to know how the new bacteria are named.
Yeah. Okay, ISROensis, Fred Hoyle, and Autobiogenesis. That is how the bacteria were named. Okay, the homology was something like 99%. But one new to the science was Cyanobacteria hoyle. Okay,
correct.
And there are about 12 more bacteria. Many unknown, undetected were fungi. Of course, there should be a continuity in the program to follow up, and those people who have done at the Hyderabad lab and Pune lab, everybody has retired now. So I think it ended up there.
Anyway, we tried to incorporate second payload, could not succeed because of the reluctance from people involved there at that particular point of time. But hats off to Dr. Dutt, because He was the project director, alone, single-handedly handled it for 3 full years, and was very strong in biology, and he rose to the level of an engineer, did the job, of course. He put it correctly. Vedacharam came to help us, and that was a great addition to us at that time. Of course, I was a party to many reviews with the payloads. It was good, and I'm sure the students appreciated the presentation, and it was a fresh air from that. So far, we're talking about only space. Now, nearer the Earth, doing experiment with balloons is another experiment. I do remember 1973 May, we conducted a balloon experiment using Aryabhatta. Aryabhatta model was carried by a balloon. We did the X-ray astronomy payload in a spinning satellite from a balloon altitude. For me, participating in 1973 and then again 2005, 30 years later, was a great experience. Because I do believe balloons are not optimally utilized by us. Maybe more and more experiments can be conducted, and with available facility at TAFR again, it is available today. I think we can do far better experiment like what Dr. Dutt mentioned.
Thank you, sir. Thank you very much.
Thank you, Vijay.
Sir, we have a very preeminent scientist from SAC, Dr. Parihar.
Parihar, yes, Parihar.
Parihar, sir, namaskar. You have to switch on mic. Mic. Unmute, sir. हम तो धन्य हो साहब, आप you could able to listen to us.
Parihar ji,
I think
something went wrong. But sir, anything? Vijay Bhatt?
Yeah, I mean,
thanks to you are acknowledged by that in the beginning.
Thanks to your Space Club and Dr. Dutt in special, I think it was a learning thing for me, because 2005, I had heard about this, but I didn't bother much about this at that time. So it is such a nice thing. I didn't know it. So much is there in that. I mean, I wish I should have learned that time. So today was a learning thing for me. It was such a nice thing for our students here in US Space Club to hear on this topic, which is of their great interest nowadays, for students. And only I have one doubt, which may be a very layman doubt from the point of view of this domain. It is nothing to do with astrobiology. I have some doubt. You talked of this balloon landing in Bidar, Gulbarga, near Gulbarga.
Yes, sir.
If I'm not right, this balloon flight, it is not a controlled landing. It is not a controlled landing, and location perhaps you would get through GPS, if I am not wrong.
Yes, onboard GPS.
All through the flight.
Yeah.
Now, how is it, is it traced? How is it? Tracing is fine through GPS. But when it lands, it is not controlled. So with a 625 kg payload, something which is landing somewhere.
Somebody dies, I would have died there.
Issues related to it. I just want to understand how you address those issues.
Yeah, we will tell you, sir. We'll tell you. Sir, in fact, the trajectory is continuously monitored. The GPS is displayed through a GPS control board. Okay. Yeah. Now, thanks to Geeta Vardhan, the Madhavan Nair sir has requested her to arrange for an helicopter. The moment I will say that my balloon is cut, because it will take about almost 1 hour to descend. So at least half an hour before, if I can tell the Coimbatore, I was given the confidential number, and immediately I could contact, then the helicopter was already there in the Shamirpet. Yeah. Okay. The moment the balloon was commanded to the balloon burst. Actually, no, balloon is cut through pyro cutters, and eventually the payload is suspended with a gravity fall. The gravity fall is very, very high, and we use a parachute. The parachute is used at 12 kilometers because we wanted to delay the landing, and the impact on the ground is very much controlled. Okay. You have asked Bidar, how do I know and all? As we told you, the high altitude balloon, when it is being launched, prior to the launch, 2-3 days before, we try to understand what is the upper atmospheric winds and which side they are blowing, and what is the strength of the winds. If the strength of the winds are higher than 250 kilometers per hour, we abandoned that flight. In fact, we did that. On 20th April, we wanted to fly. We abandoned that. And ultimately, we have did it on 25th. Because, we also need aircraft clearances, NOTAMs, everything. So many aircrafts at the time, airport was very dense, Begumpet, that was a problem. So we have taken care, and yes, luckily it has not fallen on anybody's house or anybody's thing. So far, it's not happening. Otherwise, it is it will raise to certain kind of conflicts with the people.
Okay.
But thanks to Gita Vardhan, she could arrange the helicopter, and we were tired, and you have to trace it. You asked me, how do I trace it? It's a big paratroop with a red color and all fluorescent color. I was looking through the window, open window. I was very bold enough. At 2 kilometers altitude, it is flying. I was telling the pilot, please take a merry-go-round 2-3 times to confirm that is the balloon payload. Parachute would fall somewhere nearby 1 kilometer somewhere the payload. So that took about 15 minutes to perambulate that area. And thanks to the very, very efficient pilot, where recent our combined command Army General expired. Same aircraft, similar aircraft, only the Indian Air Force has provided. Thanks to ISRO because of their magnanimity and blessing.
Sir, I just want to add something. Maybe one of the youngsters asked about today's technology.
No. Computers have nothing to do with this.
No. Sorry, you don't assume that I will talk of computers. Why do you assume like that?
You are a computer man.
Yeah, I know you assumed it. It is not so. But today, such flights, if they take place, if there is relevance, then there may be some systems possible which are miniaturized in terms of mass, which will provide a little bit of controls in such a way that the landing doesn't take place in an area where some harmful things can happen. So these kind of things are possible nowadays. That is my guess. That is what I wanted to give.
Sir, in fact, balloon AB3, in fact, the third experiment we wanted to do, Dr. Vijay Rahman can watch for me. We have from 625, we wanted to do it 25 kg. And some of your lab instrumentation people from VSSC, ISAC, we contacted. Almost 1 year dialogue was there. And ultimately, of course, that was for various unknown reasons that could not take place because of the manpower and all that.
Okay, sir. Sorry for taking slightly more time. Kutti, sir. Sorry.
Parihar sir, please come forward. Please come forward.
Mike.
Your mic is off. Your mic is off. Please put on the mic.
My best friend, very eminent agricultural scientist.
Parihar sir. Mic on kariye.
Mic on karna.
Is it okay now?
Yes.
Okay, so thank you, CBS, for really very illuminating talk. You took us back in time, and that amount of details which you have given about the flight, really, even today I got so excited as we were when we heard and read about that experiment. And thank you, Dr. Vijay Rahman, for encouraging that kind of things in that era when ISRO was indulging into many of this kind of activities from the Earth observation point of view, which were not directly connected, but we were indulging. And that was the spirit of ISRO, which made it different from rest of the organizations in the country. I wish ISRO continues with the same legacy in indulging into near frontier fields of science and technology. Wish you all the best, my young colleagues. You are in that era where you can think of those things which were initiated and done to some extent, but there are many more things which are undone. So I would urge my young colleagues, students, to indulge into some of these things and approach the ISRO and various organizations to think of this kind of missions. Once again, congratulations, CBS, and nice to see you after a long time.
Thank you, Dr. Parihar.
Thank you, Parihar Saab. It's so nice. And one thing, out of these comments, one thing come out. ISRO is known to the student community for various things like rocket, satellite, student satellite, so many things of that variety. But when it comes to the final benefits to the man on the ground. The people involved in that and the project concerned are some of the people who here, headed by Dr. Vijay Rahman. Earth Observation Office of ISRO was the place where people grounded on the Earth, and whatever space could derive benefit for people, it was achieved in this office. And many of the scientists and people involved in that, I had a day-to-day contact with them because I lived amongst them in the same building and the same space and the same canteen eating thing. So I enjoyed that life in my ISRO tenure, and I salute people like Vijay Rahman, Ganesh Raj, CBS Dutt, and a variety of people who contributed so greatly. And some of you must read the book written by S.K. Das, IAS person, Touching Lives. He has very vividly explained, how a common man, the so-called Aam Aadmi, get the benefits of space. So today's CBS has taken us back to some of those things. Thank you so much. Thank you, Parihar Saab.
Thank you.
Thank you, sir. Thank you very much to every one of you.
Anything more, Sayuj?
No, sir. All questions are over.
And I think my friend Ganesh Raj wanted to ask here.
No, Dabji, I don't want to ask anything because your presentation as usual was fantastic. Because you have taken back to all this bacteria era. In fact, we used to joke it is Dutt bacteria. I remember in canteen those times. And certainly it's a new era. In fact, we have studied, like as it was stromatolite, early life, what we studied earlier. A lot of scope is there, but I find very less research is done in this field. It is not focused much. A stray research is done here and there, not really focusing on going further. I don't know, maybe because people believe God created Earth and God created human beings or everything. That's why it is not going at, I feel. You take anywhere, it is simply some research and some paper comes, no conclusive evidence. I feel sometimes very sad. Science is stuck in this. That's one my very frank view. Because recently we have on a Geo group also, we have discussed about this evolution of Earth and evolution of life. Recently we had a big discussion for about 1 week with the latest developments. So I find it is same what I studied some 30 years back. Not much change. So maybe youngsters, I feel, like Sayuj or the team should do something and come out with more evidence, whether it is extraterrestrial life came to Earth, or is it the biogenic origin, abiogenetic origin, chemical origin? What is that? I request youngsters to think and come out with a good solution. That's all my suggestion and comments, sir. Thank you, Dutt. Fantastic presentation.
Thank you, dear.
Parihar also, and Bhatt Saab also, and Kutti Saab, of course, as usual, very promoting this activity. I told in the afternoon also. He has done a wonderful job by creating a very, very dynamic group of space enthusiastic students in Calicut Center, no doubt, like Epicenter, and surrounding districts. A great service by you, sir. All the best, sir. Thank you.
Thank you, Ganesh Rai, sir.
Mahadevan, would you like to say
Yes, sir. So, Dr. Datta, I think I had some interaction when we were in the ground station, I think quite some time back. So that's the only time I remember that we had some interaction. But this is a different area, and we really learned very basic things also from this lecture, because you have taken us through from beginning to the end. This is a very great area, and to know about the evolution and the way the living organisms are being found at different altitudes and all those things are extremely intelligent and very good for research as far as the students are concerned. So it was a excellent lecture, entirely different than what we have been normally listening in such these places. So thank you, Dr. Datta, about this thing.
Thank you very much, sir.
So I hope.
Papan, are you there?
Kutti, I want to add one
Please,
I'm
please, sir. It is
Ganesh Raj mentioned about.
Papan, just 1 second. 1 second, Papan.
Ganesh Raj mentioned about the canteen talks about Datta, not Dutt, Dutt bacteria and all that. In fact, it's not Dutt bacteria they talked about. They named it Dutteria. Instead of bacteria, they said Dutteria. So it is a lot of jokes. Interesting comment those days.
Acha, Papan is here, that inverted S-curve of the balloon. Professor Rao has teared him into pieces. We all suffered almost for 15 days. Papan sir?
I am not expert in balloon flights, but we worked together for a present.
The Professor Rao's input now.
Yeah, we had a struggle for 15 days to make a paper for Professor Rao. How the human invasion of the forest in Brazil we presented it. Yeah, coming to the students, we have many students who are interested in this particular subject you have been talking about, that is origin of life, origin of earth, etc. Very many of them. But what the students might have understood from Dattaji's lecture, it is lecture I have no comparison, because he has started from biology to chemistry to physics to satellite or balloon making. It is a very complex field. Okay. So to understand the origin of life, origin of earth, you have to understand biology, chemistry, and you should know how to measure it, how to use the technology. May not be the as cloud computing may be useful later, but technology you must explore. It is very complex. It's a very, very open field, challenging field. As Ganesh Raj put, we have not moved 1 inch from last 30 years. It is up to you. It's a challenging field. You must take it up. I thank Dattaji for opening up this field for the students community, as well as I joined to study more about this, and you educated me. Thank you, sir.
Thank you very much. Thank you very much.
Well, I found Papan missing in this presence here. So I called him around after 15 minutes of that talk. So he said, I am not able to access. So,
well, good is I was no struggling with the cloud computing. I am struggling.
I know you are interested and you want to come, but seeing you missing, I called you.
Google was giving me problem. You are out of space. But finally it allowed me in. But I missed about 1 or 5 minutes of your initial lecture, Sataji, but I think I can extrapolate.
But in that 5 minutes, I told the problems we faced together.
I can extrapolate.
One strange thing I see here is, most of the people here, Jairam, Parihar, we had Shivaraman and few others from SAC. So even Papan and Jairam also originated from SAC, if I remember correctly. PRL, correct. I mean Ahmedabad, PRL. So Papan is PRL. So it's a very nice crowd here, from ISRO side.
Yeah, I wanted to comment on that. Yes, there's a crowd of ISROians here.
Okay, thank you very much. Anything more, Sayuj?
No, sir. All doubts are cleared, I guess. No more questions from students' side.
So, Shajil Marsh?
Okay. Thank you, Sayuj, for wonderfully handling these questions session. You are our strength. Sayuj, Krishnendu, Varun, and Kumar, these are the people who are actually running this platform. We are only the supporters, and we are so happy to see your involvement. And we could see that with Ganesh Raj sir, Padmanabhan sir, and Jayaraman sir, and Mahadevan sir is continuously supporting our mission to the students, and we don't feel any kind of alienation from any other sources. So we get strengthened with our mind, and it would be great boon for the students also. Then we are going to the conclusion, and we'll of this today's wonderful, interesting program. And today's vote of thanks is from the part of ULCC is done by usually done by T. Damodaran sir. Is not available now, and we have entrusted that duty to Krishnendu, our student coordinator, and I invite Krishnendu to express vote of thanks. And before that, Varun will make the announcement on the UL Space quiz.
Sir, thank you, sir. Thank you for that, sir, for an exciting presentation. I